Oh, okay. Thank you. Good morning. It's a great pleasure to be here to present our work. But first, I would like to start by saying thank you so much to HFSP for supporting this work between different institutions, and one of them is in Africa. So I think it's one of the rare research programs under HFSP, which is covering across planets and involving Africa. We've had a great team, and I just want to start by thanking the key PIs. Dr. Lucy Ochola, she's in the crowd, managing our team in Kenya, and my mentor and a great friend, Professor Paul Robinson, and his leadership. We are running the program, and also Dr. Iwan Bai, he's also here. He's supported us, he's an engineer, and he has worked with us to run some of the engineering work that I will present here today. So the story I want to share is about migration, which is very puzzling, the migration of schistosoma eggs. And before I talk a little bit about why we decided to pursue this topic, I just want to put into perspective the significance of the work. So as we all know, schistosomiasis is a major neglected tropical disease. It's affecting over 200 million people worldwide. And you can see from the graph that the concentration is on the African continent. And so we believe that by tackling some of these issues, we'll be contributing to improved disease management and also leading to eradication. So this is work which is just starting, but I think the ultimate goal will lead to the last sentence below here. So yes, I want to take you a little bit back to the biology of this very amazing life cycle here. The story begins in the aquatic life cycle where the snails release the Saccharia, and they infect humans. They migrate to the liver, they multiply in the liver, and the adult worms, they move to the mesenteric vasculature where the schistosoma eggs are laid, and this is where the puzzle begins. So they are laid in the vasculature, but how do they find their way out? Apparently they do, and they get out. The metacidia get back into the snail, and this old life cycle continues. For billions of years, this has been going on, moving between two different hosts and also causing a lot of disease problems. So our puzzle, the question that we set to answer in this project was, how do the eggs migrate? Where they're laid, and they get out to continue the life cycle. So a lot of biologists have studied this, and I just want to illustrate the migration process as we understand it now. So it starts with when the eggs are laid, and the egg, you will see up there the immune cells. I don't know whether my pointer is working, if you could allow me to. Okay. All right. Okay, there we go. Okay, seems to be working, sorry. So you will notice we got the egg laid, and immediately we have the endothelial cells and a lot of other cells will come to encapsulate the egg, and they will cross the endothelium, the first barrier that they cross, and then we have the process of granuloma formation where the macrophages, a lot more T cells and other immune cells will come and form the granuloma around the egg. And somehow the granuloma is able to move. So is it polarity issue? What is the force driving this? That is the question. And eventually it will move past the epithelial and find itself in the lumen. And the story continues. So the question we wanted to ask is how they manage to go through different tissues, pass through the barriers, and get into the lumen. So this is a very difficult question, because remember the eggs, they don't have any appendages, no mechanism of self-locomotion. So they have to rely on the host, just as I've explained, relying on the host to move across the different tissues. So the hypothesis that we came up with, and this is something that we're still tackling, we looked at tissue mechanics, and we thought about who may be the players that could be driving this process forward. So the hypothesis that we came up with is, when you look at the recruitment phase, where the egg is surrounded by different host cells, immune cells, we kind of focused on this person here called the fibroblast. And we thought the fibroblast, because of their ability to be contractile, and they are well known to change, they can be immune stimulated to move, to change from fibroblasts to myofibroblasts. And the myofibroblasts are very, very contractile. They actually deposit a lot of extracellular cell matrix. And so we thought this process, there would be a key player in the whole tissue mechanics that eventually drive forward the granuloma. So here I'm showing the assembly process where the ECM deposition by the fibroblast would cause some sort of stiffness differences between back and forth, the back and front, I would say. And that force imbalance would create a force vector that might drive this forward. So this is the hypothesis that we set to try to understand. So it involves a lot of steps. First of all, we have to be able to quantify the forces, and we are able to have this assembly set up in an in vitro system. So we've been working the baby steps, and today I would like to share some of the progresses we've made and some of the challenges that we've faced. And so this is work still in great progress. So I just want to say that we are not alone in being interested in this process. This is a study by a group in the US, University of California, San Diego. They focused on how the cells cross the endothelial barrier. And what they noticed is when the eggs are laid, the VEC, the vascular endothelial cells, will try to wrap themselves around an egg. I think this is due to the immune stimulation. The eggs are known to be immunogenic. And when you look at this, they kind of did a study and found that mature and immature, or if you kill the eggs, you will have the different, the spines, or the tails that they wrap around them will be different. And they also kind of looked at, they made an in vitro system with the vascular endothelial monolayer and looked at the stresses that the cells, that the endothelial cells are actually producing around the eggs. And what they found is that actually there's a bit of polarity. If you look at this, you will see that there's the compressive forces, and also there's some tensile forces, that there's a bit of polarity in the force vector around an egg. So, and actually they did some analysis to see that the mature, immature, or dead eggs, there would be some differences in the forces around an egg. So they kind of gave us a hint that our hypothesis of the force unbalance may be actually something contributing to this whole unique motion. So well, just as I said, we decided now let's integrate in vitro models with in vivo models and try to elucidate the whole migration motion based on tissue mechanics. And what we did was to build a team. Just as I said at the beginning, that we have a team at Purdue University and a team in Kenya. So we split our roles, that at Purdue we will try to come up with an in vitro system that could simplify or make it possible for us to decipher this complex process. And in Kenya, their role as great biologists would be to try to decipher the same motion in the in vivo setup. So yes, the team is multidisciplinary, so we started, I'm going to start with the in vitro system work, where we were trying to build a microphysiological system that would capture the whole motion that I'm showing schematically here, with motion starting from the capillary, crossing the stroma, going all the way to the lumen. And we said the best way is to try to use the hydrogels. So we wanted to set up a system that we can observe microscopically and also try to map the forces that are generated during this migration process. So we came up with a system that we can position on a microscopic lens and then do the imaging using the bead assay to track how the movements generate forces in the matrix. So just to highlight some of the things that we've done, to be able to do imaging while assessing forces is not very easy. So we had to come up with systems and ways to do this. And one of the things that we did was to use the engineering skill. This is work that Dr. Bai actually supported. So some very smart engineering students used the 3D printer and a low-cost device that actually could permit in situ imaging, and we could compare the performance with the rheometers so that we could tune the stiffness of the gel, because stiffness is known to actually influence the whole migration process, because the migration actually depends on cells probing the matrix, depending on the matrix stiffness. So we did develop this system. We could position this, mount it on a microscope, and we could actually do the imaging. And you will see that we could get really nice pictures. And so the system was working. Then we had to understand, is it capable of giving us the stiffness of the gel? Because stiffness is important for us to quantify the forces that are developed when the migration process moves. So this is we did compare the 3D printed device with a standard rheometer, and we chained the gel stiffness of a wide range, and we could actually determine that within the linear range, we could actually determine the stiffness, and we were able to come up with a stiffness that is a little bit physiological to the extent of about 10 kilopascals that was able to continue the work. So after that, we went to the next step of trying to create this bead assay system. The whole idea here is, as movement occurs over the gel, we should be able to track the vectors that tells us how the movement or the forces are developed. So this is a very old trick where you have fluorescent beads embedded in a gel, and whenever movement occurs, there will be deformation, and when we track the deformation, we are able to quantify the forces. So for this, also we used the same device, and we could deform the gel. You can see how we fabricated the gel. We just embed the beads in the gel in polyacrylamide gel, and then we could deform the gel using the device under a microscope, and we could quantify the forces that are developed when movement occurs within the gel itself. So here I'm just showing you some of the clips. You will see the gel embedded with the beads, and then you will see this. We can set it on a microscope, a built-in microscope. We can do continuous imaging when under deformation. And actually, we could use PIV. It's a technique of particle imaging velocimetry, very much used in biophysics, to track the movements of each bead. And from this displacement vector, we are able to quantify the force vector based on the stiffness that we already measured using the device. So we went on to actually do actual work, and we could actually get the vectors. But this is where the long story starts, because doing the gel, doing the vector, the first thing, this was part of a very nice engineering. We could work on this, we could do this. And then the toughest part was now doing the biology, getting the cells to be happy on the gel, and to produce the forces that we wanted or we expected them to do, this was where the challenge started. So we seeded fibroblasts on this, and we continued to assess that, but the fibroblast would not be happy on the gel. So we are still continuing to track their motion, but that is proving to be really, really tough. And getting the eggs to move them around on this gel is something they're still working on. I see my time is running up, so I just want to clarify that the in vivo research is also going on, and I can't stop without saying that they have really done a lot of work to try to come up with the models that capture this. It's a long process which involves going to the field, collecting the snails, coming to the lab, growing them in the lab, and they are able to infect the mice and get the mice to replicate the disease condition. They've been able to go into tissue imaging and prove that at least when infected, the granuloma would form as expected, and you can see very beautiful images there. So right now they're doing what we call longitudinal assay, starting from week 1 to week 14, and assessing how the kinetics of this granuloma formation and also the cytokines are changing. So we are looking at from the infection phase to the acute phase to the chronic phase, and looking at how cytokines are changing through this whole process. And they're using the multiplex bead assay. Our smart PhD student, Martin, has been able to quantify some of the cytokines that are involved in this whole infection process. And you will see that we have different mice tracked over a long period of time, and we're looking at how those cytokines are changing during the infection phase from week 1 all the way to week 14, and we can continue all the way to week 20. So we believe this time-wise will give us a much clearer picture of what immune cells are moving at what stage, and how the granuloma itself is forming, and the picture will become complete as we continue this exciting project to understand the whole migration issue. So I think I've been able to tell you that we've set up the in vitro system. We're still working the biology, and the in vivo work is still ongoing. So it's a long, winding road to success. And just to say thank you so much to the students who worked on this, and also to say that we had a lot of fun, and thanks to HFSP for the support.
Thank you.
And thank you so much.
Thank you very much. Kennedy, since we are